Good afternoon. Today we are going to talk about the differences in tissue organization among C3, C4, and CAM plants. In this presentation, we will go over how these plants optimize photosynthesis under different environmental conditions and the leaf structures they have developed and the role of diffusion as described by Fick's law in reducing photorespiration. These are some real-world examples of C3, C4, and CAM plants, where C3 plants contain vast majority of plants, which is about 85%, and includes common plants such as wheat, potato, and rice. C4 plants are less common, that covers about 3% of all species, and examples are corn and sugarcane. And at the end, we have CAM plants, which covers about 7% of all species and usually located in deserts. As an example, we have pineapples and cactus. In this slide, we will discuss the photosynthetic strategies of these plants. C3 plants. These are the most common where CO2 is directly fixed by the enzyme Rubisco in the Calvin cycle. Although efficient under moderate conditions, C3 plants become less effective in high temperatures because of increased photorespiration. C4 plants. In contrast, C4 plants use a two-stage process. Initially, CO2 is captured into a 4-carbon compound in the mesophyll cells. The compound is then shuttled to the bundle sheath cells, where the Calvin cycle takes place in an environment with high CO2 concentration. This spatial separation reduces photorespiration. CAM plants. Instead of a spatial separation, CAM plants use temporal separation. They open their stomata at night to fix CO2 into organic acids, storing it until daylight when it's released for a Calvin cycle. This method minimizes water loss and helps curb photorespiration under arid conditions. Comparing leaf structures in T3s, in C4, C3, C4, and CAM plants. C3 plant leaves. The leaf structure is relatively uniform, optimized primarily for light capture and gas exchange without any specialized anatomy to combat photorespiration. C4 plant leaves. These leaves exhibit what's known as Cran's anatomy, where mesophyll cells surrounded the bundle sheath cells. This circular arrangement creates a microenvironment with elevated CO2 levels around Rubisco, effectively reducing photorespiration. CAM plant leaves. Often succulent in nature, CAM plants have tissues that not only store water but also accommodate this storage of CO2 in the form of organic acids. This design is vital for survival in extremely dry climates. The distinct leaf structures clearly mirror the plant's specialized adaptations, emphasizing how structure and function are intertwined. Let's relate these differences with the principle of diffusion with Fick's law. Fick's law states that the rate of diffusion is directly related to the concentration gradient and the surface area available for exchange. In C4 plants, the Cran's anatomy boosts the CO2 concentration within the bundle sheath cells, creating a strong gradient that drives rapid CO2 diffusion right where it is needed, minimizing unwanted oxygen interactions. CAM plants exploit a similar principle over time by collecting CO2 at night and releasing it gradually during the day. These plans ensure an optimal gradient for efficient diffusion during the Calvin cycle, all while conserving precious water. These, these strategies showcase how the laws of physics are harnessed by biological systems to enhance survival and efficiency. To summarize, C3 plants operate with a straightforward photosynthetic mechanism but are prone to photorespiration under stress. C4 plants have evolved specialized leaf structures, crans anatomy, that creates a high CO2 microenvironment, significantly reduce photorespiration. Can plants manage their gas exchange by separating CO2 uptake and its utilization temporarily, which conserves water and maintains efficiency in harsh environments? This adaptation not only highlights the diversity of life but also offer insights into how plants might be optimized for future agricultural practices in a changing climate. Thank you for your attention.